Hi guys, welcome if you're joining me. Sorry, I'm just setting up at the moment. Oh, it's been a tiresome day. Hi Callum, how are you? How are things going? Right, let's have a look. Let's have a look. Let's have a look. There. Welcome guys if you are joining me. If you have questions, please do ask away. Sorry, I'm just trying to set up. You're good, thank you. Busy week. Yeah, it's been one of those busy weeks. I have been so in my zone. I've been building. It's getting so close. Look at the state behind me. It's so close to being ready now that I have done a biological molecules, biological membranes, enzymes. I'm almost finished the workbook. So yeah, so it is so close to being done that I am like, oh, I will be so glad when it is done. It's just going to take a bit of one last little bit of time, but it is so close. And I'm very excited about that. Very, very excited. I've been busy with it's my daughter's birthday. So we have been busy doing that. So that has been fun, but also tiring. And so there we go. If you want to join me, please do come and join me. If you have any questions, please do ask away. And yeah, it's exciting times. Oh, I've just lost my pen. There we go. There we go. Right. Do we have any questions tonight? If anyone is interested, I am running an Easter revision intensive masterclass. It's a whole day on the Tuesday, the Wednesday, or the Thursday. You can attend in the end of March, beginning of April. For anyone who is looking for some intensive full day studying with me, I am going to go through all the key areas where students lose marks, showing you walkthroughs of how to answer the questions. Also, I've created your own notebook with practice questions for you to do as well. So there will be ones that are past exam questions, but there are ones that I have actually built for helping you to improve. And obviously you get access to all the resources at the end with the recordings from the day. So if you are interested, then please do check out my website, ukbiotutor.com, where you can find how to book onto it. And don't forget, if you are wanting to attend, you can get a discount if you type in Easter 26. So I am offering a discount for you if you put in Easter 26. But if there are any topic areas that people would like to actually cover tonight, then please do ask away. Otherwise, I will just get going with doing some questions and going through how to answer those questions. So let me have a look. What have we not done in a while? What have we not done? So let me see. The last one we were doing the 23 paper, I believe. What did I do then? Have a quick look. So last paper. And that one. Hi, Carol. How are you? I'm extremely tired. My youngest got up at a very silly o'clock this morning. I think she was excited. And she decided that that would be a perfect time to wake mummy and daddy up. Well, actually, to wake mummy up. And so I have been awake for a long time, which means I'm a little bit on a go slow right now, guys. So I do apologize on that front. But I'm going to go through, I believe we are 2023 paper 1 I was looking at, and we were going through the questions on, there we go. If I just open them up, I can then start looking at the questions. So please do feel free to ask if you have any questions that you want to ask. We're checking to see if you were live, as I was hoping to ask some questions, but I can wait. Oh yeah, sorry, I did pass your message on, by the way, so I don't really know the answer to that, and I'm not going to pretend like I do. So yeah, but I did pass it on. If they haven't responded, then they might not know the answer, but it was a interesting evening, shall we say, and I've been working. Oh, okay. Can you send me a message? That might work better. Right. What are we doing? I don't even know tonight. What am I doing tonight? What do we want to do? Nobody seems to be asking any questions. Everyone's just watching to see what does she actually cover. Either that or there is something wrong with the live chat on YouTube. So guys, if you are trying to write a comment and it's not working, then I don't know why. Please do keep trying. I will try and respond. I think I found paper. Have I found the paper? Here we go. We've got to. Yes, so we were talking about the roots, and we were looking at the IAA concentration last lesson that I did this. And I find this really fascinating. Whenever they give you a picture that looks like this, so let's go. There we go. So when you have a picture that looks like this one, or I say a picture, what I mean is a table of results, it's important to note what are they actually telling you with this type of set of results. So they are giving you the concentration of IAA, and IAA is obviously, it's working. Nice, thank you for putting a comment there. I can't actually see anything apart from your comment saying it's working, so thank you, I do appreciate it. Hi, thank you. Yes. So guys, if you do have questions, if you are in my live chat and you have questions about biology, please do ask away, because this is why I do these sessions, so that I can answer any questions that you may have and try and fill those knowledge gaps that you may potentially be struggling with. But let's look at this question. So one student's results are shown below. So they were talking about the roots growing and the shoots growing. And when a seed germinates, so if I have a seed, what we would expect is that the roots are going to grow down and the shoots are going to grow upwards. So when they germinate, when they have been activated, and they get activated by the water, which is going to come through into the seed. And when the water comes into the seed, that will result in the activation within the seed to germinate. Now, to germinate, it requires a hormone, germination, and that hormone is gibberellin. And what's going to happen is that gibberellin is going to activate the cells. And inside the cells, what you're going to get is the switching on of genes. And the switching on of genes is going to be useful in breaking down the starch. So to break down the starch, what we need is we need the production of our proteins, more specifically our enzymes, amylase. And the amylase will break down the starch into the monomers, which is the glucose. And what will then happen is that glucose can either be used for respiration, it can be used for growth, i.e. be converted into cellulose for production of the cell wall. And right now I'm not actually I'm not actually answering the question. I'm just going through the information. Is this important for NEET? So if you're not educated at the moment and you're not interested in the A-level biology, then no, this might not be important for you. However, if you are interested in keeping on top of your education for when you do return to school, then yes, this could be of benefit for you. So it really just depends what you're doing and what your interests are. One of the things that I would say is sometimes you can get hung up on it being only doing the stuff which is necessary to pass if it's an exam that you're sitting, whereas actually sometimes finding your interest allows you to find a passion you didn't know was there. So yeah, it's one of those, it really depends for you. I hope that answers your question though. Right, so we're talking about germination, we're talking about seeds, we're talking about them being activated, we're talking about this growth. For example, the cells are going to go through their cell cycle, and we know the cell cycle goes from G1 to S to G2 and then through mitosis, and then from mitosis to cytokinesis. All of these are energy demanding processes. So to grow the roots, to grow the shoots, what you're going to need to have is a increase in the number of cells, as well as the increase in the cell's size, so the cell elongation. What is cytokines? So cytokines are chemical messengers which are released in response to a invasion by a pathogen. Are you talking about cytokinins, which are to do with the plants and promoting the plants going through mitotic division? Or are you talking about the cytokines, which are your chemical messengers? So do make sure when you are looking at the word that they've given you that they have asked about, is it cytokines or is it cytokinins? And it's that slight bit of a difference that's going to be what they're actually asking you to describe. And it's one of the things that I struggle with my spelling, and I struggle with my dyslexia, and so therefore because of that I end up having to really take the time to sound out what it is that I'm trying to say. So do make sure that you go through that. Can I compare lysosomes to lysozymes? So a lysosome is a vesicle, so a phospholipid bilayer which is encompassing, that's not the word I want to use. It is holding inside it the lysozymes, and the lysozymes are the enzymes that are going to be used for hydrolytic reactions. So lysosome is the vesicle containing the lysozymes. So basically we've got a little compartment inside the cell which contains these enzymes. We don't want these enzymes breaking down the cell itself, so we've put them into a little room, and that room is the lysosome. The lysozyme is the enzyme that's going to break it down. You are very, very welcome. Right, can you please go over cloning? Can you be a bit more specific for me, Izzy? When you say cloning, I'm 100% happy to go through cloning, because that's what I want from you guys. I want these questions where you're not sure, and I will try and fill them up. So when we talk about cloning, there is the natural cloning, and then there's the artificial cloning. So one thing I would say, if I was doing this and I was doing my revision, as I'm doing with you guys right now, is I would split it down into, are we talking about plants or are we talking about animals? And then are we talking about it being natural, or are we talking about it being artificial? And the other thing I would say to you guys, cloning in plants. Perfect. Thank you, Izzy. One of the things I would say to you, and I have a laugh with my students as we are doing this, is that my writing, I know when I'm rushing, can look really squiggly and sometimes hard to read. And I watch these other teachers out there who are doing their videos, and I think, how on earth are you able to make your writing so neat and so lovely to view? I would go so slow if I made my writing really neat. But I would say to you, if you are struggling to read my writing on this screen, then understand that your writing that the examiner sees on their screen is going to be very distorted as well. So if you are someone who struggles with your writing in that it's not as neat as it could be, then I would say please do take the time to make sure that the examiner will be able to read what you've written, because if we can't, we're not going to spend hours on your answers. We will obviously try to decipher it, but if we can't, if there's just no way of deciphering it, then we're not going to be able to give you the marking points. Okay, let's look at this though. If you want and you're looking for tips for OCR A, please do pop across to my channel where I'm currently live at the moment answering questions on my board. We're currently doing cloning in plants. So if I look at natural ways in which plants can clone. So we have just finished in the winter months. And so what you should know is that in the winter months, your plants have either died out or they have actually had storage organs hidden underground. So for example, one of the key ones to note is the likes of potatoes. So potatoes are amazing. If you like potatoes, if you like them, you can have them boiled, you can have them fried, you can have them baked. It really just depends how you want to have your potatoes. But we're not talking about cooking them here. We're talking about how the plant is actually going to use them for a way in which they can then produce a new plant. So we know through the process of translocation, so this is where we bring in our transporting plants, and I know this is not what you asked, but I will always link the topic areas together, synoptic links that you can find. And if you can think about biology in a bigger picture, the easier it will become, and you'll be able to pull that knowledge forward. So because of translocation, we know that our sugars, our complex organic molecules, can be moved from our source to our sink. And the potato plant is really quite clever. And what it does is it will store some sugar in its stems, and you'll end up with this swollen part of it, which is the potato. So when you have this swollen part, which is a storage of starch, what you will have is meristematic stems. So this is actually called a tuber, and on the tuber, whenever you've left the potato for long enough, it will start to actually grow what's known as the eye of the potato. That is where you will find meristematic cells, and that is where the plant will start to grow and produce the parent plant from those meristematic cells. So it's actually going to grow when the conditions get better. So it has stored the storage organ, has stored the starch ready, just like with a seed. When you have a plant that produces a seed, that seed has got a storage of starch inside it. So starch is our polysaccharide, which can be broken down into, as I was saying before, the glucose molecules, and the glucose can then be used for lots of different things within the plant. So when we're talking about cloning, we're talking about genetically identical plants being produced. So over winter, what's going to happen is it's going to produce these tubers, these storage organs, and once the weather becomes good again, once it starts to get to that point where the conditions are right, it will start to promote growth, and that is where you have the eye starting to come out of the tuber. So tubers is one way in which it can happen. I'm going to take away the animals part because you said you wanted plants. The other way could be, for example, a strawberry plant. So the strawberry plant, what it does is it actually produces what's known as a runner. And the runner is a horizontal stem. And what happens is when that drops down to the ground, what will happen is it will start to produce roots in the ground, and then you'll start to get your strawberry plant growing from there. So a runner is a plant. It is an adapted stem, and it will, and I'm like sitting here going, la 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 la, here's my lovely diagrams. And I'm like, I actually have a notebook on this which has some beautiful diagrams because I went through and I made them, and it's my most recent notebook. So why am I not showing you that? Oh my gosh, why do I not do this? It's crazy. I'm like, oh yeah, I forgot. It's there. So here we go. Let's just take this now. And there we go. Let's put this out. So here we go. Oh, wrong way. There we go. There's my potatoes, and I'm showing you the eyes of the potato, which is in my study guide that you can actually buy. You could have the garlic with the bulb. So if you look at another form of natural cloning is where you have a garlic bulb. Now, I don't know if you've ever actually looked at the garlic bulb, but what you actually have when you've got a garlic, it's really fascinating, is you've got another specialized underground part of the stem. And so it's a storage organ, and it's the same with like daffodils. The daffodils right now are blooming. It's lovely. They were storage organs under the ground, specialized storage organs under the ground, and they waited until the conditions were right before they started to bloom and come out of the ground. And so therefore what we've got is natural forms of cloning. So you could say the same with an onion. Let me just move this one across. So you've got the onion as well is another example. And the important part is within these, what you have are these lateral buds, which are going to actually start to grow. So these are the parts which have the meristematic cells. And meristematic cells obviously are the ones that can go through mitotic division, and they are the ones that will be producing the new cells, and those cells will then need to elongate. So that's if we're looking at the natural cloning. So you've got your runners, you've got your bulbs, you've got your tubers. So different ways in which the plants can actually clone naturally what they need to. And then if we start to look at what are the ways in which we can have the artificial cloning, well, one of the ways that we do artificial cloning is through the process of taking a cutting. So it's important when we do this that you are looking at did it again. That's not what I meant to do. Let's try that again. Let's skim through it. And again. So we're going to cut the plant, and we need to cut it from a non-flowering region of the stem. And the reason it's non-flowering is because that is where you're going to find active meristematic tissue. That is where you're going to have hormonal levels that actually promote the growth. And it's really interesting, Skylar, you said down here, don't forget, guys, auxin, APD, inhibits lateral bud growth. It's important that if we remove the apical meristem from our cutting and we only have the lateral bud, you've not got anything that's inhibiting it. And so it's important to note where you're taking it from, the fact that you are taking it from an area that has these meristematic tissues. The fact that you're going to cut it at a slant, you're going to allow for this larger surface area for water uptake. And going back to module 3, transport in plants, understanding the fact that the cohesive property, adhesive property of water will allow water to move up through the xylem, and so therefore you want the water to continue to go up, and so you obviously want to increase the surface area for that water uptake, and so that's why you want to cut it diagonally, and also you want to reduce the risk of rotting, and it will help it to successfully root because you need the roots to grow. Pramila, I'm answering the question because Izzy asked about cloning in plants right now, and someone else has asked about detoxification. So I am going to try, if I've got enough time, to go through that as well. But I just want to finish this. I am basically answering the questions that anyone may have to do with any of the topic areas, to be honest. So the other thing is about dipping it in rooting powder. Now, if they're going to ask a question on this, they could potentially, like they have in the past, given you an example of what someone else has done and asked you how you would improve it with that method, or if they're asking you about why something happened. So they could, for example, give you a set of results, which was somebody took a cutting from a plant, they dipped it in rooting powder, and they didn't dip it in rooting powder. Then they measured the root growth after a set amount of time, and they compared, and they want to see if there was a statistical difference, significant difference between one or the other. So it really depends what they decide this year to go down the route of what they're going to ask. So it's about not just memorizing, well, you do this and you do this and you do this and you do this. That's good if you have that knowledge, but can you then apply it to a random example that they may decide to give you in the exam? So there is the cuttings that you can take, or the other one that we would want to talk about is the fact that we would do, I'm just moving this across. For anyone who is interested, you can actually buy my study guides which have this in. So I did do one, my most recent one, before my humongous one that I'm in the process of building right now. That one, it was on the cloning topic area. So if you are struggling, then please do check it out. And I also, randomly, the more you bulk buy, the cheaper it becomes. So do check that out because it may be of interest to you. So if we're going to do the micropropagation, if we are going to take our plants and we are this time going to not take a cutting, but we're going to do tissue culture instead. So if we're going to do tissue culture and micropropagation, I don't know if this is going to actually work. Oh, it might work. Are you going to work for me? No, there we go. So what we're going to do, we're going to take a tissue culture. Now, to take a tissue culture, what you will want to do is you want to remove part of the plant. And so when you remove part of the plant, you are going to scrape some of those meristematic cells. You want the cells that are going to be able to go through mitotic division. And so if we want to do that, we want to ensure that we are only taking the places where they are more likely to have that meristematic growth. So we know it's unspecialized. We know that it's going to be able to divide by mitosis and then differentiate. And therefore, we want to remove it from either the tip of the root or the tip of a shoot, or we may want to remove it from a bud. The key thing is we don't want any contamination. So we need to ensure that we actually do sterilize what we are removing. So we need to sterilize it. We need to put it into a sterile medium, a culture medium that's providing the necessary nutrients that it will require. So it's important that you understand when you're sterilizing it, you're going to use a sterile scalpel to remove the explant. You're going to wipe the surfaces with ethanol to remove any contamination. All these things that they will want you, and I do feel like aseptic technique is going to come up quite big this year. So if you are someone who has only just come across my profile or my videos, you'll know that every so often I get an idea of this is, and that is one that's sticking in my head. There's a few that I feel that they are going to go more heavy on, but I would say to you, be aware that biology can take you in any direction they want to go. So it's not good enough to just memorize one topic area. You have to see how it all interlinks. So the aseptic side of things, if you don't want contamination, you are going to want to ensure that you have removed only the cells you want, and then you're going to make sure everything else is sterile. You can either grow it in a liquid nutrient broth or on a solid agar medium. So in this diagram, I'm using a solid agar medium. And what we're going to do is we're going to transfer that explant across. Then what we're going to do is we are going to allow it to actually start to do the things that it needs to do. And what's important is that we know that it needs to go through the process of cell division. But before it can go through cell division, it needs to be able to replicate the DNA. If we're going from a cell or a couple of cells and we are allowing them to divide, they need to do DNA replication in S phase before they can go through that mitotic division. So therefore, we need to provide them with the nutrients such as amino acids, such as phosphates, such as glucose. So what's the amino acids needed for? Protein synthesis. What's the phosphates needed for? DNA and ATP synthesis. What's the glucose needed for? Respiration. So all these different things, depending on which part of the topic area they want to assess you on. So it's important you know why these things are useful for the plant. So as long as we provide them with the right nutrients, what's then going to happen is they will be able to go through that division and they'll divide to produce a mass of identical undifferentiated cells. Now that is called a callus. That callus can then be split apart. So we can take this callus, we can break it down into smaller, I don't even know, is it calluses? But smaller masses. And if we break them down into smaller masses, what we're then going to do is we're going to keep doing that to repeat and get desired clones through taking only cells. But we have to give them the right conditions. So we then have to monitor them. We then have to let them grow. We then have to plant them and see what happens from there onwards. So it is important to note when you are looking at cloning plants, yes, there are the natural vegetative processes of the tubers, so the tubers, the runners, the bulbs, the I can't even say the word tonight. Wow, my brain. Rhizospheres. What you basically need to know is if they give you any of those examples, it's not just about the fact that that is one named example. They may ask you to explain how that links to something else, like the breakdown of the starches, like the transport of the sucrose down to the sink for that storage, or the release from the starch stores, because that's now the source, to the sink, which would be the cells that are growing at the roots and the shoots. So it really just depends. So Izzy, I hope that's been helpful on that front for you. But if you are interested, please do check out my cloning study guide, because all the information is on there, but it is available on my website if you are interested. Right, Kevin, if you are still in here and you are still interested in the detoxification of the liver, that's an interesting one. So detoxification, whenever you see the prefix D, when we talk about D, whether it's deamination, whether it is decarboxylation, whether it is dehydrogenation, DE normally as a prefix would mean the removal. So detoxification is the removal of a toxin. So some form of toxin is going to be broken down so that it's no longer there. Is micropropagation a type of cloning? Yes, it's a type of artificial cloning in plants. Micro means using the small side of it. Micro, small, you're only taking a small sample, so you're using the tissue culture technique for micropropagation. Micro, small, making lots of copies of it, so getting a large quantity of the exact same plant from that small sample that you took. So I hope that helps, Izzy. Back to the detoxification then. If we were looking, and they tend to go down the route, a lot of the likes of your hydrogen peroxide detoxifying because of catalase. So if we're going to detoxify it, we would therefore convert it into our water and oxygen. So 2 hydrogen peroxides are going to give us 2 waters, and we are going to then get 1 oxygen. You are very welcome, Izzy. You are very welcome. Thank you for joining me and watching. So if we're going to detoxify, we need the cell to contain things like an enzyme, which is catalase. So that would be one way in which we could detoxify the hydrogen peroxide that is being produced. Maybe it's to do with ethanol. So if we are of age and we have consumed alcohol in the form of ethanol, we therefore would need the right enzymes to break that down into a less toxic state. And so the role of the liver, because all the blood is circulating first, once you absorb from your small intestine, it's going to go to the liver via the hepatic portal vein. And so therefore any toxins that you may have consumed are going to be taken there and then be detoxified. Did you not see my Chidima? I did see your question. I am above you though, because I've not got to your point yet. Which bit were you saying? Can I ask how much do we need to know about natural clones in plants, tubers, runs, etc.? Like as far as is that the question you're on about? As far as you need to know the example, but you also need to understand the example. It's not going to be good enough for you just to know the names, because it's about you understanding why it's a natural way of cloning and how the plant can then grow from that natural clone through the process of mitotic division, differentiation, through the process of potentially becoming a specific part of the plant, like where the xylem and the phloem goes, It could be to do with the hormones. It really just depends where they're going to go with it. It's never just the names. It's about it how biology is that bigger picture. So it's quite a difficult one to know when it comes to the cloning of how far they're going to go into it. Now, they did do a lot on animal cloning this year, so that would make me think that it's probably going to be maybe a more likely a small marker or an application question. So yeah, but linking back to the detoxification, hydrogen peroxide is one that they always love to throw in there. Catalase breaks it down. The fact you've got what do you produce, how do you actually use and set up a practical for that? So it's about the fact of how would you control, what would you control, what's your independent variable, what's your dependent variable, what are your values for your independent variable. So if I was going to, for example, look at differing concentrations of hydrogen peroxide and how they affect the rate of the catalase, that could be something that I'm changing. So if I'm changing the hydrogen peroxide concentration, I need to therefore keep the temperature, I need to keep the pH the same, the temperature the same, the liver pieces, I need to make sure I've got the same mass so that I have the same enzyme concentration. So it really depends on the practical. What if I'm, instead of using liver, I'm going to use potato? Is that a better way? Is that an improvement? Because a liver is really difficult. If you've ever tried to cut up liver, it's really difficult to cut without damaging it. So therefore, are you affecting its efficiency by the way you have cut it? Whereas if you were to use potato, would that be better because you can control the surface area in a better way? So again, it could be that they're asking you about the practical investigations, or they're asking you about module 1 and assessing your ability to evaluate a practical and how could you improve it. Now in my example, I have a measuring cylinder upside down. I've got a trough where my trough is filled with water. I have that there is water filled in my cylinder that's upside down, and I have a delivery tube which is going, and therefore the bubbles of oxygen are going to be collected, and as they rise up they will displace the water, and that displacement is going to be the measure of that volume of gas. Is that the right way to do it? Should I be using this upside down measuring cylinder, or should I be using a gas syringe, for example? Is that a better piece of equipment for me to use? So it really does depend what they decide they're going to ask the question on, as to what they want you to talk about. And as I was saying before about the ethanol, if we're going to use the ethanol is obviously a biotechnology topic area, in the fact that if we've got anaerobic respiration in yeast, that anaerobic respiration will result in alcoholic fermentation. Alcoholic fermentation, we have that pyruvate, the end product of glycolysis, can be converted into ethanol and then ethanol. And so if we use that ethanol, i.e. in our brewing, we can make beer, we can make alcohol, we can make these fruity cocktails if we really want. And so that would be looking at the biotechnology topic area and linking it to the liver. Well, if I drink and I consume the alcohol, how does my body detoxify it? So once I've taken it in, once I've consumed it, I then will need to break it back down into the ethanol. And to do that, I need alcohol dehydrogenase. So to do a reversible reaction, I need an enzyme for that. And so this enzyme is going to take the ethanol and convert it into ethanol. Now, if you looked at anaerobic respiration and alcoholic fermentation, you should know that pyruvate is decarboxylated removing a carbon dioxide. So we're going to have some form of decarboxylase enzyme involved. That is going to remove the carbon dioxide to give us ethanol. The ethanol can then be reduced to ethanol by accepting the hydrogens from the reduced NAD. Therefore, that is going to oxidize the coenzyme NAD, and that can go back. Well, if I'm going to go the opposite way, I'm going to need to have something to accept those electrons. So again, thinking about how they may switch it to see, can you actually apply the knowledge that you have about how the processes work? They know, for some of you, you are just going to memorize these topics, and you can go like glucose to pyruvate, pyruvate to ethanol, ethanol to ethanol, but not actually understand what is going on. And if you don't understand what's going on, when they throw that curveball at you in the question, when it's an unseen novel idea, you then like, and you're stuck. Whereas if you take a step back and go, okay, what do I know? Well, to go that way, I needed an enzyme. To go back, I need another enzyme. What enzyme am I going to use? Oh, and they tend to give you the name of these enzymes in the stem of the question. So it's really important you look at what they've said in the stem of the question to see if there's any hints there for you. And then you've got the fact that the ethanol changing to ethanol and then changing to ethanoate, and then that can either be converted into fatty acids or can be used in cellular respiration depending on what is needed. And so it's important that you know that it really depends which metabolic pathway it goes down as to what is going to be the stem of the question and where they want you to go. Okay, so it is all down to whatever they decide this year they want to assess you on. Does that answer your question, Kevin, if you're still here? They're kind of like the two things for detoxification. I guess the only other thing that I would say would be the deamination. But that's more formation of urea when it comes to liver. So hold on, let me just have a quick look and remind myself, because my brain has been so in this module 2 study guide that I feel like my head is filled with that. And I would say to you, like, even though I have done this and taught this in so many ways, formation of urea is slightly different. So yeah, they're the two that I would say are the main parts. Yes, it does. Thank you. You are very welcome. Just because I'm on a roll with it, just to finish this off, if you haven't watched my video on the liver, the deamination of excess amino acids. So obviously deamination, removal. We're going to remove the amine group, and that amine group will get converted into ammonia. And if it's converted into ammonia, this is a toxic substance. So in high concentrations, that would become an issue. So what tends to happen is the ornithine cycle will convert the ammonia into urea. Urea is only toxic in high quantities. So therefore, we are changing it into a less toxic substance, and then we're excreting that. Now, because that is a nitrogenous waste, when we excrete that as urine, the microorganisms, and I actually really do have a feeling, again, here's a little snippet for you. I have a feeling that nitrogen cycle is coming up, and it depends which way they want to go with the nitrogen cycle. But animals producing urea being excreted, it can then be broken down by decomposers, and that can produce a ammonia or ammonium ions, and those ammonium ions can then be converted into nitrites and then nitrates, and then that can be used by the plants. So they could go down that route if they decide that they want to assess your understanding of the production of the urea, the excretion, and how that's useful within an ecosystem. I'm just like throwing these things out there, but that's the way I could see a question coming up. If it doesn't come up, I can see it coming up in the future. Right, where did I get to? So I hope that's answered yours. Skylar, give me a second and I'll bring in epistasis. Because I've still got kidneys on it is fab. I'm concerned they swell up with starch and then. So the starch is the storage molecule. The key thing is starch is the storage of energy inside the plant. So energy is required for so many things. So if you store the starch, that's therefore going to mean that that starch can be broken down. The fact that it's 1 to 4 and 1 to 6 glycosidic bonds means that it's a good storage molecule. It's compact, it's insoluble, it doesn't affect the water potential, and so that is therefore going to be a source when it starts to grow. So the starch becomes the source when there is no photosynthetic tissue to do photosynthesis. So when the plant above ground, for example, with the tubers, when the plant above ground has died off, the tuber sits there and waiting and then becomes the source to provide the necessary glucose for breaking it down. It was mostly this and the histology of the liver that I found quite confusing, so thank you very much. You are very welcome, Kevin. Histology is difficult. In the last 2 years, they have given hard questions on the histology of the liver. So I'm assuming you have potentially had them in mocks or you've seen them in some of the past papers. But I would say, as long as you're looking at the lumen size of the blood vessel, as long as you, the key thing to be able to identify first off is a sinusoid. If you can identify the sinusoid, then you look at the lumen of the vessels behind and in front of the sinusoid. You can then gauge where you have your hepatic artery and your hepatic vein, and then you're looking at the two before to work out whether it's a hepatic artery or a hepatic portal vein, and the lumen size will be the hint that gives it away. So the questions, when they give you histology, it's normally pretty obvious which one's which. There will be something that allows you to figure it out if you know what to look at. So when I say it will be pretty obvious, it's only pretty obvious if you know your content, if you know your topic area. If you've just memorized hepatic portal vein, hepatic artery leads down the sinusoid to the hepatic vein, when they throw that question at you, which I want to say it was 2024's multiple choice question, because I feel like it was 2024's. It may have been 2020. No, I don't think it was 2025. I think it was 2024's. Yeah, it was 2024's question, because I remember after they sat the paper, I sat down and looked at it, and I sat there for ages umming and ahhing over what it was with the right answer, and it was to do with the lumen size. And it was interesting because me and another teacher were disagreeing as to what the right answer was. Obviously I was right, I mean, come on, but I don't always get it right. I was lucky in that moment I got it right, and I would never get 100% in the multiple choice, and I would never get 100% in these papers. And that doesn't mean I don't know my content. It doesn't mean I don't know the stuff that I teach. It doesn't mean that I'm not good at my job. It just means that sometimes I don't think in the same way that the examiners want me to think, and I won't get all those marks. And so I'm okay with that. And I have gone through my teaching degree. I've gone through my processes of understanding that I work in a different way and that my brain works in a different way. And so therefore I would say to you, try your best not to get hung up if you don't get 100%, or if you don't get all the marks, or if you don't get the grades right now in the mocks, in the practice tests, in the past papers that you're doing. That's not what's important right now. What's important right now is, one, that you're trying, two, that you're identifying what you don't know, and you're going and looking up and researching and watching a video or doing a flashcard or reading a textbook or making notes, whatever works for you, that you are doing that, rather than burying your head in the sand and thinking, well, what's the point? I'm never going to get there because I don't have enough time. Someone did ask me on here, have I got enough time to improve to get to that end goal of what I want to get. And absolutely 100% you do. You have time, but you have to be smart with your time. You have to do what's needed to get you to understand the bits you don't know. You will have bits of in your brain that you are unaware that you know that what you're doing. And what I mean by that is, I remember when I was doing my PGCE and we had to have these professional studies sessions, and it was always talking about things like being unconsciously competent and then consciously incompetent. And I've got to think this one through now because there's so many different ways of saying this. Give me 1 second. I have to write it down because otherwise I won't be able to write it down. Just the last thing of the carbon skeleton can be used in different ways. And if they are going to ask about the formation of urea, they don't want you to know the ornithine cycle, but they have asked this question. The key thing is it's an enzyme controlled reaction known as the ornithine cycle. And what they were actually looking at in a question like this was they were looking at, you understand that you're going across membranes, and do you know ATP gets broken down into cyclic AMP? But what I wanted to go and talk about here, if I can remember this rightly, so 1 second. So there are 4 stages to this. So if we've got 4 boxes, you can be uncompetent, and you can be competent. You can be conscious, and you can be unconscious. And so it's talking about the fact that for some people you can be consciously, not incompetent. I'm saying the wrong words here. Oh my gosh, my brain is so not with it. You can be consciously incompetent. So if you're consciously incompetent, you're aware that you're not very good at something. You can be consciously competent at something. You can be unconsciously incompetent, and you can be unconsciously competent. At different parts of your time throughout studying, you will be in different boxes within this table. And it's an interesting one. I mean, I'm going basically 20 odd years ago now when I was training that this was what we were being taught. But it was talking about the fact that when you first start to drive, you're aware that you're not very good at it. You're aware that you're not very good at it. And then you become aware that you're getting good at it. So you are aware that you're not very good at it. So you are consciously incompetent. You're aware that you're not very good at it. You then become consciously competent when you practice lots and lots of time. You become aware that you're good at it. Sometimes you can actually be unconsciously competent, in the sense of sometimes I can drive from here to somewhere else. Let's say I've driven for an hour and a half, and I'm not actually sure how I got there, but I got there because I wasn't thinking about what I was doing. My brain was somewhere else. I love driving because for me, that's the time with having ADHD, I need something else going on. Otherwise I can become a bit like, whereas when I'm driving, because I'm physically doing something, my brain becomes so clear that I get such great ideas when I'm driving. And I used to drive up to Scotland all the time to go visit my parents a long time ago before the girls came along, and it became a lot more challenging. But yeah, I used to love driving because it would be when I would have my perfect, in my mind, my perfect ideas, but the ideas would just flow and they would come to me and I'd be like, this is so amazing. I don't remember why I started going down this route, but it was basically to say to you guys that sometimes, I do remember now, sometimes I have students who don't believe they know the right answer. Because they don't believe they know the right answer, they're like, oh, well, it can't be this. And it was sitting there all along, but they weren't aware that they actually had it in them to give the right answer. And so I would always say to you, if you have a gut feeling or a gut answer that you want to give to a question, put it down, okay? Come away from it, carry on, come back and review the question that you've done. Come back and review your answer. You should have enough time at the end of the exam, hopefully. You should have enough time to do a quick read through of the questions that you struggled with. So one of the other bits of advice I would say to you, as you're going through the paper, mark the questions that you found a bit more challenging, so you know to review that one. You didn't feel confident, you wrote an answer, but just double check, have I written the right thing down? But yeah, that would be my advice. I'm sorry I didn't manage to cover epistasis, Skylar, but what I would say, Skylar, if you look at my playlist for patterns of inheritance, I have got a video on epistasis, and I feel like I did epistasis 2 weeks ago in here on my live, so it should be in my live videos. If it wasn't last week, it was the week before, but I definitely did do a session on epistasis, so please do check that one out. But guys, I am going to finish now because I am extremely tired. As I said before, I've been up since 4 o'clock this morning. Actually, that's a lie. 4:30. 4:30 was when I got woken up. And there's only so much my brain can take up until that point. So I want to say thank you for joining me. Thank you for those of you who are continuing to watch my videos, continuing to share and like and comment. It really does mean the world to me. I want to make more videos for you. I really do. But at the moment, I'm prioritizing my study guides, and I'm prioritizing my revision masterclass that I'm doing in the Easter break, because these are the ways in which I make the money that means that I can still do this. And so obviously I appreciate you guys for coming along. I appreciate you for watching and liking and sharing, and the more that you do, the more I can still keep doing these things for you for free. So thank you for your detailed explanation, and thank you for oh, you're very welcome, Kevin. Do you know what, I remember what it was like to be a student, and I also know what it's like to not be able to afford tutoring, and it makes me happy to know that I am still helping students by doing these sessions, and I do enjoy it. I love teaching. I'm a bit tired tonight. I'm a bit tired most Wednesday nights, to be honest. I teach 3 hours before I jump on here. So it's a long evening, and I taught earlier today as well. But I do love doing this. I love helping you, and I love knowing that maybe one of my videos will just make it click for you. And that is why I do what I do. And so, yeah, thank you for coming along. And obviously, the more questions you guys ask, the more I can actually build these resources around, because it's not just about you guys coming here now. It's lovely, and I really appreciate it, but it's also those of you who are now watching this at a later date and are finding it useful, because you also were struggling with that question. So many students are scared to ask the questions in class in front of their peers because they think they're going to be judged, they think they're going to be asked or ridiculed, or it's this horrible experience of when you are surrounded. Like, schooling system is so unique an experience because you are surrounded by people of the same age, and therefore you are judging yourself against your peers, and actually you are so different. You should never judge yourself against anyone else. It's about you and you making progress, and the progress that you make, however big or small that may be, is progress, and that's what you have to take forward with you. There will be some setbacks, there will be some things that you're just not going to be as quick at picking up, and that's where you put the effort in. Some topics that you just don't like, that you hate, in fact, don't avoid them, do them. And if you want, I promise you, if you are anything like me, neurodiverse, and hate revising, hate homework, hate studying, But at the same time, like to learn, then another thing you should do is check out aiqchat.com, a revision platform that my partner and I built, because actually that is very much built on a way of guiding you and helping you to fill those knowledge gaps. So if you are struggling, I would advise checking that out, and it's free for the first 7 days. So if you don't like it after using it, you cancel subscription. You don't get charged anything. So you've got 1 week's free use of it, and that will therefore help you to gain knowledge in those topic areas that you hate. So yeah, check it out. Guys, thank you so much, and thank you for all the likes if you have been with me on YouTube, and thank you for all the likes if you've been with me on TikTok, and have a good evening, everybody.